Our new interdisciplinary project between biology, computing and thinking about wider sustainability issues is called Wildlife Genetics, and I'm just going to show you how to run through this project with a class. So I've built it around this idea from the STEM education and training strategy, thinking a little bit more, how can we link other disciplines together? How can we focus on that and pull those different aspects of activities out for our learners? But also, this activity is ultimately grounded in the biology curriculum, and I think particularly suits the advanced higher curriculum where we're thinking about phylogenetics. But also we could look at this within the higher curricula, whether it's human biology or just higher biology itself, around genomics, where it starts to think about bioinformatics. And arguably you could maybe pull this down to National 5 as a way of introducing DNA sequences and how we can use that to learn more about an organism, but it would probably be quite challenging at National 5 level. So the project starts with our aim, which is placing the learner as a conservation scientist who's gone out to a particular national park in Taiwan, and they've collected different biological samples. When they've gone back to the lab, they've analysed that, they've extracted the DNA and obtained the sequence. And they now have to try and find out which type of organism did that sample come from. And we're going to use a new platform that's been developed through the University of Dundee with various collaborators called GeoView. So it starts by getting the learners to have a wee look on the map. Where exactly is this national park? Because that comes back into this project later. So they'll start by clicking on that link there to download the sequence alignment, which has already been done. If you struggle with that link at all, just send me an email and I'll send the link directly to you. But when you click on that, you should get into a file and we can download that. It's a very specific type of file. So we click download. And because I already have GeoView downloaded as a desktop application, I get this little icon here to show that it's basically a FASTA file. If you're not going to go down that route and you're going to use GeoView within a web browser, which a lot of schools might do, you won't have an icon here. Download it and then you can upload within the web browser itself, and then it recognises the file format. Okay, so that's us downloaded that. So we move to step 2 and it says, I'm going to just use the desktop application, but all the instructions are there if you're using the web browser. So all we're going to do now is find our sequence alignment, and I'm going to double click it and it's going to launch GeoView. A range of different windows here, and the main window for this, it depends what you're loading up. So we've only got the multiple sequence alignment window, and you can see up here, very small, but we've got our unknown sequence. This is the sequence that we have obtained from this national park, and we've aligned it to 4 other different sequences. And the first part of the instruction says just to manually scan the different sequences that you have, and you have this consensus bar that shows you how similar the sequences are, and wherever there's a gap, that's telling you that there's a difference in those sequences there. So largely you can see that they're very similar. We're now going to select a colour, and we're going to colour by nucleotide. And so that allows all the adenines, thymines, etc. to be of a particular colour, and it allows you to much more quickly see where there's differences. So the first part is really to get the learners to have a look manually by eye and to make some kind of judgment about the similarity of these sequences. And really the point there is that this is really difficult. It's really laborious. It's very error prone. It would take you a long time, especially if you had to compare our unknown sequence to many, many more sequences. So how can we make that an easier job using bioinformatics? So the next step looks at what's called pairwise alignment. So to do that, you're going to left click on the first one, hold down the control key and select the second one that you want to do a pairwise alignment. Calculate pairwise alignment and you get a new window. So that will show you in this window, if you scroll, the pairwise alignment between the 2 different sequences, and then right at the bottom it will show you your percentage identity. So that's out of all the different bases that we've got, what percentage of them are identical in the two sequences? And that's 88.66%. And what you would do is your learners might have a table of some kind that they've got in their jotters or on their electronic jotter, whatever that they have, and they'll just start plugging these values in here. So if they looked at the unknown and Panthera Leo, they'll type in their answer here for what that pairwise alignment is. And similarly, you can just shut that window down. Then you could do unknown, hold down control and go to the next one, calculate pairwise alignment, and you get your new value here for that. Once you've done all that and you've populated your table, you're going to move on to the next part, which, first of all, step 7, that's going to allow you basically to figure out where that DNA sequence has come from. So what species was it that was in that national park? And then you're going to look at phylogenetics. And this part here is just directly from the Advanced Higher Course spec. So it puts it into the context of this activity. And then we're going to actually carry out a phylogenetic analysis. So let's have a look at how we will do that. So across all of those different species, what you're probably going to want to do now is avoid using the unknown, because now you'll know which organism the unknown sequence actually is from. So for the purposes of just showing you, I'm going to include these 4 here, and we're going to calculate and do calculate tree or PCA. And then in this window here, you're going to do average. It's quite annoying how small this is. But this bit here, if you click there, you can see it is just DNA that you need. On the web browser, it looks a bit better than that. And then you do calculate. And up comes your tree. Again, really small, but you can copy and paste and make that look a little bit better in PowerPoint or something. So you get this tree here, which allows you to make some conclusions and some analyses around how related different species are, and come back to that course spec again. So there's a couple of questions in this document for you, for your learners to follow up at that point. Then the sort of tail end of this is to really then go back to the organism that you found. That was the species that we found in the national park. Go into the red list of threatened species and have a discussion around, was that significant? Now this is fictitious, this is all made up, but I guess it's based on something that could happen. So they're going to try and work out if that was a significant finding or not. And then if they wanted to take it further, there's some further suggestions about how they could actually get involved with some citizen science as well. And the tail end of this is just looking at the JALView platform itself. And if you click on this little bit here, it will take you to the JALView website, which has got lots and lots of other activities for you to get involved with as well.